you. So yes, in my group we are mainly working with different aspects of gene regulation, but we're also running a core facility for bioinformatics, meaning that we are doing quite a lot of other things. So I could also talk about bacteria degrading oil in oil reservoirs, and I could talk about a lot of cancer research we are doing, and so on. But instead I decided to do some advertising. So this is advertising for a tool developed not mainly by me, but by a former PhD student from my group who has moved to a different university, but we still have collaboration, and he has developed this tool. But before I start presenting the tool, I will give some sort of background and motivation for the tool. Fortunately, I can skip 50% of that because we have had very good presentations of some of the background. But I will still say a little bit about it because I will tell about it from a slightly different perspective. So very much of what I'm going to talk about is somehow related to genome annotation, labeling the different parts of the genomes with features that we know something about. And that can be quite challenging, as you have seen some examples of. But what type of features are then relevant or interesting to accumulate knowledge about? Of course, the obvious question is, where are the genes? And that turns out to be a more tricky question than you maybe are aware of. Of course, we have the protein-coding genes and the non-protein coding genes, but we have seen examples of defining the gene start. Where does a gene actually start? That can be challenging, in particular because that depends on the cell type, whereas we try to sort of annotate this, at least to some extent, according to a reference genome. If you include all different cell types, then we draw in data, so we try to show the most representative examples and so on. The distinction between protein coding and non-protein coding is also getting more and more fuzzy, because it turns out that some of the non-coding, meaning non-protein coding RNAs actually code for proteins, often very short proteins that are very difficult to detect by prediction methods. You have to do it experimentally to find out that they really code for something, and that really has a function. And then there are features like the CTG islands. We are looking for features that are conserved across species, because that tells us something about how important this region is. But of course, the conservation pattern in a protein coding gene is different from the conservation pattern in a non-protein coding gene. Because here in the non-protein coding gene, what is conserved is maybe the folding of the RNA, the complementarity. And therefore, it's the complementarity that is conserved. And that is difficult to detect with some of the standard methods for alignment and so on. Repeats is of course important. Repeats are very frequent in the genome. Sometimes they can be seen as mainly noise, but some of the repeats are important for defining binding sites for transcription factors and so on. And in the sequencing, mapping these repeats can be quite tricky. I will come back to some of these features further on. Variation, SNPs, how your genome is different from the reference genome is of course important, because that tells us something about differences with respect to binding of transcription factors, mutations in proteins and so on. Histone modification, we have talked a lot about that, promoters and enhancers, binding sites for transcription factors. All these are features, and even more features are available that we want to annotate in the genome. And of course, you need to visualize the genome using a browser of some kind. UCSC and Ensemble are the most commonly used browsers, and I will come a little bit back to that as well. So, what I'm going to talk about is genome tracks. That is some kind of feature that we can represent in a linear feature along the genome. We can also call it an annotation track. And a very common way of representing such track is simply as a bed file like this, where we have the chromosome, the start position, the end position, what feature it is, and maybe some intensity of the feature. How well is it conserved, how high is the intensity, if it is an expression value, things like that. And of course, this also introduces a challenge when you are doing bioinformatics, because the numbering system here is obviously according to one specific reference genome. So if you then upgrade your reference genome, you also have to upgrade all your reference data, which can be quite a burden sometimes to have to do. And then you visualize it. So this is maybe not a typical example, but this is an example from the UCC genome browser. We have indicated, in this case, most of the gene predictions that are available in the UCC dataset. And you see that they basically tell the same story, but there are some differences. And some of these differences are real, because there are differences depending on which cell type did you use when you did the experiment and so on. And some of them are sort of artifacts because the prediction methods are not optimal. They make mistakes sometimes. And then you have conservation patterns and things like that. So these are typical examples of tracks that you would like to compare in data analysis. So let's see a little bit more about the different features that are relevant in this context. Genomic conservation is obviously interesting. And it's basically just doing a multiple alignment of cell genomes and looking at the conservation pattern in this multiple alignment. And the assumption is that they have conserved these regions because they are functionally important. And we see a very relevant example of this here. Here we have a specific gene. Here we have the intron-exon structure. So we have the exons here. And we see peaks corresponding to the exons. So they tend to be conserved. And the intron regions, they are less conserved. But we also see examples of some conservation in the introns. And in particular, if you have included more of the promoter region here, we would obviously see conservation there, corresponding to, for example, binding sites for transcription factors. So this can be used to some extent to identify interesting regions in the gene. Another track that I mentioned briefly that is very interesting to include is, of course, single nucleotide polymorphisms. So here I focus on changes that affect a single position. We have a lot of other types of variation that is also relevant, particularly if you go into cancer. But the SNPs are the sort of the easy ones to handle as a fact. And most of this variation will not have any effect. It's just noise in a way, because it doesn't affect the property that is important for the function of that gene or the gene product. But when we do population studies, the variants that have an effect are the ones that are interesting. And very often, when you do, for example, a cancer project, you focus on the ones that occur in the protein-coded regions, the exons. So it's very common to just pull down the exons and do sequence of them, and then you see a difference, and then you assume that this difference, or sometimes this difference leads to a change in the protein-coded sequence. So you get a mutation in the protein, and that mutation can affect the folding of the protein, or the binding of ligand, or something like that. But you should be aware that there are also so-called silent mutations that have an effect, even though it's silent in the sense that it doesn't affect the protein code. The protein is the same. But there have been published at least one example where you have a mutation, or the normal situation is that you have a rare codon at that position. So you have a codon that is not very frequent. Therefore, it takes a little bit of extra time before the tRNA recognizes that site and the translation continues. And that pausing is important for the folding of the protein. So if you mutate it into a more common codon, so that the translation goes faster, then the protein misfolds. So at least one example has been published of that. I included a reference for it because I wasn't originally planning to mention it, but now since I can skip fast about some other things, I mentioned it because I think it's a very interesting example. Repeat regions, I won't say much about that, but as you know, hopefully, repeats are extremely common in the human genome. There is a lot of repeats. And they may actually cause some problems. It's, at least if you are doing sequencing technologies that are fairly short reads, then to uniquely map a repeat to its correct position in the genome can be very challenging. So therefore, you often see in at least old sequencing data, you see so-called repeat collapse, meaning that you group together all the reads for that repeat in one sort of representative repeat. And therefore you underestimate the amount of repeats and length of repeats. With longer with sequencing technologies that make longer reads, that is improved, but it's still a challenge. CPG islands has also been, or the CPGs has been mentioned. And you should be aware that although there is so-called definition of what a CPG island is, different groups use slightly different definitions. So different tracks from different groups may vary a little bit with respect to exactly how they define a CPG island. But basically, it's a part of the genome that has a very high density of CPGs. For example, this definition here, is 200 base pairs. The percentage of DC should be about 50%, and observed expected CPG ratio should be about 60%. Then it's defined as a CPG island. And this is annotated, for example, in the UCC genome browser as a separate track that you can use to visualize. Histone modifications, this has been mentioned quite a lot and in good detail, so I won't go much into it. You have already heard about the histone tails that are modified in different ways, so that you can use that to identify active genes, repressed genes, virulent domains, and so on. So some things here I can skip quite rapidly over. And then you can accumulate different types of tracks into a visual display. So here we have the gene, here we have the CPG island, we have the SNPs, here we have repeats, predicted by repeat masters, so that you can filter out repeats for analysis if you want to do that. Here you have histone modification tract, in this case layered for different cell types of the HD27 escalation tract, so that you see basically the gene starts where it starts. Also here, transition start site peaks integrated, total counts and max counts. You have DNase clusters, transition factor chip aggregated so that we can expand it later on and look at different transition factors, conservation. So you have a lot of different types of information that you can now analyze visually.
What is total counts?
This one? Yeah. Maybe you know better about that because this is phantom data.
I just asked what is total counts.
Yeah, so I believe this is the max value that is added. Here
I guess if you have several case.
Oh,
yeah.
I guess it's if you have several different samples together, put into one track, then you can either take the sum of all the samples or you just take the highest sum.
Yeah, I think that is what has been done. And this is the same basically, but with the ensemble browser. I have to admit I'm using mainly use C browser. Okay, so this is sort of background, the motivation for what we're doing. We have a lot of different tracks like this. Here we have a large number of CTCF tracks for different cell types and so on. There are different methylation tracks and so on. But how can we say something reasonable about how are these tracks correlated? How should they be compared? What type of story do they tell? How can we link them up to other types of information? So this is what we are going to look a little bit into. And there are different approaches for comparing tracks. And I will go very briefly into two approaches for doing it. One is a very simple approach where you test each, in this case a gene, for overlap with a property to get the matrix. And you can generate a 2x2 matrix and test the significance using something similar to the Fisher exact test or a more generalized version of that. So that is what we illustrate here in A. So you have your query set. So that can be a gene set with a specific property. For example, strongly upregulated in a disease. Then you have the background set, which can be the total set of genes. So that you know that this overlaps with that. This is the total set of genes, and these two genes here belong to the family, to the subset of strongly upregulated genes. And then you can have a property. And that property can be, for example, CPG islands, or it can be some regulatory feature that we want to test. Binding of a transcription factor, for example. And then you have here these 4 different cases. So you have this gene belongs to a family of strongly upregulated genes, and it overlaps with a feature, 1,1. This belongs to the strongly upregulated, but it does not overlap with the feature. So it's 0,1 or 1,0. This one does not belong to the strongly upregulated, it belongs to the background, but it overlaps with the feature, 0,1. This one also does not belong to the upregulated set, and it does not overlap, so it's 0,0. And then you can use this to make a 2x2 matrix and do a test. And this is a very sort of standard way of testing. Here is an example. We have method 1, method 2, class 1, class 2, corresponding to the 4 different situations that we had here. So that can be, for example, correct, incorrect, clear, and something else. And you just count how many occurrences do you have of each. And then, if you're using the proper exact test, then it's this formula here, meaning that you can compute directly the p-value for this distribution that you observe of the different cases. And of course, you don't normally compute something like this. You do it by an approximation. But still, with an approximation, you get a fairly good estimate of the p-value. And the null hypothesis you're testing is, of course, that there is no difference between these cases. So what you're testing is that the probability that you actually have something different from the null hypothesis, no difference. Then we have the other one, which is what you see here. Then you have just the query set. You don't say anything about the background, basically. And then you have the property. And then you somehow estimate the relationship between the query and the property, which can be, for example, the degree of overlap. So here you have an overlap of 0. Here you have an overlap of, let's say, 488. It's just a random number in this case. But you can estimate the amount of overlap. Or you can estimate the distance between, for example, the midpoint here and the midpoint here. That's another option. The essential idea is that you estimate the property that says something about the relationship between the query and the property you're looking at. And the query here does not need to be a gene. It can be something else. It can be a different feature. So you can estimate the relationship between the different features. And then, how do you test that? You can test that by randomization. And testing by randomization is something that is used very frequently in different contexts, in different sort of wrappings. Monte Carlo, bootstrap, permutation test, resampling, cross-validation. It's based on the same idea, but in different sort of wrappings. And the basic idea is to test the result by making a randomized version of input data and see how likely it is to get a similar or even better result than your actually measured result. So as an example, some kind of correlation test, you have data on correlation between gene expression and distance to enhancers. How significant is this correlation? And then you just do the permutations and see how often do you get the correlation that is at least as good or better than what you actually measured in your experiment. Then you get the distribution for numbers. Your measured value is here, and a few times you get something that is even better. But the majority, in this case, will be bullish. And here you can use this to estimate the p-value. And of course, how precisely or how reliably you can estimate that p-value depends on the number of permutations. So this is a compute-intensive method. Very often you start with just a few permutations and see, does it indicate that it's possible that there is some kind of correlation here? Or if not, then I just skip it. If it seems to be reasonable, then I continue with more permutations. Okay, so now I'm going to present the tool. Please feel free to ask questions because I have actually a reasonably good time since I could skip very rapidly over some of the previous stuff. But we'll go into a couple of features in addition. So the purpose of the genomic type browser is to test statistically genomic tracks and relationships between such tracks. It's written in Python, implemented into Galaxy. You already have seen the Galaxy screenshot from the previous presentation. And this is what the sort of a pure Galaxy look like. And it's a framework for wrapping and using different types of tools. So you have a lot of different tools along here, and you have the history here, and you have sort of a communication window here. And we'll come back to that. So this is what the genomic type browser looks like. Here is the link if you want to go and test it later. So you have some tools up here for doing the analysis, for processing different tracks. Here you actually have some history files for things that have been analyzed either as demonstrations of how it can be used or as part of publications. So it can actually make tracks available as a reference for your publication so that readers can go back and test this afterwards and see if they agree on the way you need the process. There are several publications on it. Sunday is my former PhD student who has developed the tool. I was not part of the first publication, I came in later, when we had made it into an even more general tool in a way. But it's still very much under development. I will come briefly back to that towards the end. So we are very eager that people actually use it and test it. Also because then you help us to find bugs. There certainly will be problems, errors, bugs in the tool like this. So if you report back, we are very happy. Not because of the bug, but because we learn to know them. One of the sort of fun presentations or projects we have been doing with this is something we have called the differential disease regulome, where we did a large-scale analysis of the genes that are associated with certain diseases. Many diseases, there are genes sort of known to be linked to that disease. So that is a huge list. And then we used a track of predicted binding sites for transcript factors. We have to admit that it's not a very good track, but that was what was available in large scale at that time we did the analysis. And then we did the correlation between the gene lists of diseases and, I think it's gene list of diseases here. So here we have all the different diseases. Here you have the different transcription factors. And then you can go in and focus on sort of the hotspots. And see here, there is a strong correlation between regulation of this transcription factor and the genes involved in this particular disease. And this became a very large map, so we actually used Google Maps to navigate it. So you can take it out in Google Maps and navigate around, highlight these spots here that you find interesting, and look for interesting correlations between prescriptive factors and diseases. Actually, quite a fun project. Okay, so what can you do with this tool? The basic idea is that you have a track of some kind, and it can handle different types of tracks. It can handle point tracks, so you know that there is something at this point, which can be, for example, a SNP from mutation analysis. And there can also be points with values, intensities. So if you have identify a very specific binding site for a transcription factor, then you can also estimate the strength of that binding site, how frequently is it occupied. What is more frequent is that you have segments. You have a region and you know there is a binding site for a transcription factor inside that region, for example. And then you can have those as just segments or as segments with a certain value. It's strong binding site or a weak binding site. Or you can have a frequency measurement that goes along the full genome. DNA bendability, for example, that is something that can be predicted. And then you can predict it along the full genome. And then you use pairs of tags and ask the biological question. Is these points occur inside these segments more frequently than you would expect by random chance? And then you use that to set up a model where you have a null model. What is sort of the normal situation, the random situation? What do you expect anyway? And then you can compare what you actually observe against what you would expect in the sort of unbiased situation, using either a Monte Carlo simulation, as I mentioned briefly, or an exact test. It's possible in some cases to use an exact test. But an exact test means that the data that you observe follow a specific distribution, normal distribution or something like that. And in reality, it turns out that biological data very rarely follow the distributions that you would want them to follow. And then you have to go to Monte Carlo. Of course, the difference is that if you are able to use an exact test, it's very fast. If you have to do Monte Carlo, it's very slow. But that's life. And then you can estimate two different types of results. You can estimate a global result, a global p-value. Over the full genome, this is significant or this is not significant. Or you can also estimate local results, because you can indicate bins that you want to compare or test individual bins. Normally, the bins are just the chromosome arms, where you can test whether it's significant in a given chromosome arm or not. But you can define these bins yourself if you want to do a different type of distribution. And this is what it looks like. I will go through some examples. This is from one of the publications. So I think I will not go very much into this one. I will come back to a more detailed example. Why should you want to use the genomic hyperparameter for these tests? One is, of course, that you have easy access to the data, because you have a lot of data now in a central repository, so you can just load the data you're interested in and do explorative data analysis. The problem with the current model is that it tends to get outdated, meaning that there comes new versions of data, new processing method that improve the processing data and so on. So in a new version that will come soon, we are implementing a sort of on-demand access. So what the library contains is pointers to the different sources of information, and then you can download the types of data that you are really interested in when you need it. And then there is a cache system, so that if the co-workers already downloaded those data, you don't need to download it directly from the original source. It's already there. But that makes a much more flexible system. One very important reason is that you have easy access to the statistical tests that you want to do. because these tests have been developed and tested by statisticians, so they know that this is done in a proper way. And it turns out that is more important than very often many realize, to be able to ensure that you use the correct test for the correct problem. Reproducibility. As I mentioned briefly, you can publish the workspace, meaning that you can create a link from your application to the workspace, so others can access it, repeat the tests with maybe some slightly different parameter settings, and verify that, yes, you did a good job. Or maybe test it in the same way for their own data. And that, of course, makes it easy for others to verify your own, including the reviewers of your manuscript. It's also easier to set up collaborative projects, because, as I said, since you can make your workspace available, you can also make it available to co-workers during the project work. And it's easy to explore data, but as you will see, you have actually access to enormous amounts of data. So you need to start with a hypothesis, with a good question. That's still the most important. Some simple examples, but first a very brief background stuff for some relevant data. Persistent factors, I understand I don't need to go very much into that, but the important thing I want to point out here is that although they recognize specific motifs, or more or less specific motifs, in the DNA sequence, the way we normally map them experimentally mean that we don't necessarily know that exact position completely. Yeah, they belong to different classes. You're probably fully aware of that. It turns out that these different classes, that is also something separate we've been doing. These different classes also tell us something about the role of the persistent factors. There are basically 3 to 4 different main types of persistent factors. We have the pioneers, the one that initiate that something happens at the promoter regions. But very often that they are important for opening up the promoter region for further persistent factors. Then we have the settlers. They come afterwards and sort of establish that this is now an open promoter region. And then we have the migrants. They can, in principle, bind almost everywhere. And as soon as there is an open region, they populate it to keep it open, to define it as a promoter region, and so on. And we have been doing a study that confirms that how they bind to the DNA to a very large extent defines their role in this stepwise process. So that's also sort of interesting. So you may want to distinguish between different types of persistent factors when doing this type of statistical analysis. You should also remember that we tend to focus on this very small part here. But many persistent factors are actually very large protein complexes. So here is the one that binds DNA and we tend to focus on. This is also extremely important for understanding the properties of a persistent factor and its role. The thing I would like to mention, and I included this because I was not sure about how much detail you would have previously about the different mapping types experimentally, but you have heard a lot of it. So I will only mention very briefly this chipset process towards the binding site. As we mentioned previously, what we actually do during this process is to get 2 peaks. And the actual binding site is in between these 2 peaks. So part of the analysis for finding the true binding site is how you statistically handle these 2 peaks in order to find the true binding site. And as you see from this, how you do the extension does not really matter that much. The important thing is that you are able to handle these 2 peaks towards a shifted peak, relative strength shifted peak, that tells you exactly where the binding site actually is. But because of this process, although you normally, many methods for mapping the binding site for a persistent factor assumes that the true binding site is in the middle here. That's not necessarily the case. So this gives you mainly a region, and you know there is at least one binding site for this transcript factor inside that region, but you don't necessarily know exactly where it is. Then you can do mapping of binding sites. You can predict where the binding sites are and try to find it, and then normally it will be close to the middle. But there also, it turns out that in most experiments where you do this mapping for transcription factors, you will always find some peaks that do not contain a motive for the transcription factor. And it's an interesting question, why? Why does that happen? Of course, some of it can be explained by protein-protein interactions. So it binds not very specifically to the DNA, but through stabilized through protein-protein interactions. But have also been a couple of publications, and it makes sense in a way, that some of this is what can be called shadow binding. In the sense that you have a promoter with transcription factors binding, and you have an enhancer coming close to the promoter. And then you do the cross-linking. And then some of the transcription factors will cross-link both to the DNA they actually bind to, but also to the DNA that comes into proximity of the transcription factor, and create sort of a shadow binding site there. And if this is a sort of stable interaction, then you will find a real density of binding sites, not real binding sites, but sort of fake binding sites in this interacting property. And that has actually been used to look for the relationship in binding sites between promoters and enhancers. So it can be quite useful if you are able to identify the weak binding sites without the motive and distinguish them from the strong binding sites with the motive. But you should be aware of that problem. So, as I said, it defines the region, but not necessarily the exact site. That is something you have to map afterwards in a sentence. I can skip this now, because you know that you're able to use hidden marker models to define the different states. And start by showing you a couple of examples using the genomic hypervisor. And I should say that if you want to find real examples that are scientifically interesting, then maybe you should go here and look at the papers that have been published and so on. The topic here, the purpose here is mainly to show how it works, and therefore I chose some sort of obvious, very simple examples. So, the first one is, as I said, fairly obvious. Test for overlap between the transcription factor, in this case me, and the histone modification for active promoters in this particular cell. And here we should expect a positive result, because we should expect a transcription factor that is active in gene regulation to bind in an active promoter. So it's sort of a trivial example, but it illustrates the different choices that we are able to make here. So we select the first track, gene regulation. We are looking at transcription factor regulation. We want to look at experimentally determined trypsin peaks. We use data from the Code Consortium of uniform processing. We use this particular cell line, this transcription factor, and this particular experiment for that transcription factor. So already the number of choices you're able to make tells you something about the amount of data that actually is available for processing. Then the second track, chromatin, from role map and genomics. This particular modification, and again we use in this particular experiment the code data for this cell line, which is the same as the cell line up here. And then we do hypothesis testing. I should mention that we can use this tool also for doing single track data analysis, meaning that we can do statistical analysis or mapping the feature of a single track. How much of the genome does it cover? What is the intensity, the max intensity, linear intensity, average intensity, things like that. But now we are comparing things. So here we have different alternatives that we can test for. We can treat these data as points. If there are segments, then we can define them as point by looking at the furthest upstream point, downstream point, or midpoint. So we can redefine them as points. do we find them at different frequencies located nearby? We can compare points to segments. Is it located inside the segment, nearby a segment, located non-uniformly inside the segment? Meaning that does it tend to be at the first part of the segment, so a last part of the segment, or close to the center of the segment? Located segments with high values, we can compare segments to segments. Located nearby, overlap, similar segment. Then we're going to look at the overlap. We are treating it as segments for persistent factor binding. As I said, we don't know the exact position, so we're treating it as a segment. And a segment corresponding to this modification. We could also use segments against valued points. Are the higher value at this point normally found inside the segments compared to the lower value points? So there are many different tests that we can. Okay, so then it explains to us how it is going to do the test. We are doing hypothesis testing. We are asking for overlap. Are this track overlapping this track more than expected by chance? Here is illustrated what it looks like. Is the overlap that we observe here larger than expected by chance? How do we treat the tracks? We use the original format in both cases as valued segments. The null model is that they do not overlap, different from what we expect by random chance. And the alternative to this null model is that they overlap more. And how do we treat this for generating the null model? Is it indicated here? How do we do the Monte Carlo force discovery rate sampling? We do it quick and rough. This is the first test. As I said, it takes time, so we want to do the quick test first. Random seed for the random sampling, just the random value. Here we can select the specific value, particularly if we do testing on the software, and it can be wise to do it so that we know that we get exactly the same result each time. But for proper analysis, we shouldn't do that. And we compare chromosome arms for the binning if we want to do that, or we can define something else. Then we say start analysis, and this is what we get back. And as we expect, yes, there may be significant overlap, but p-value is limited by few Monte Carlo samples. So we see that there is a weak evidence. This is the two tests, the alternatives that we are testing. And low p-values are evidence against the null value that are located independently, so they tend to overlap. But as I said, here we have tested very few iterations, so it's not a very precise estimate of the p-value. So here are some details about it. And the test statistics is the observed base pair overlap. And you see that this is the number of base pairs that are to overlap. Here is the number of base pairs that we observe when we do the random distribution. And we see that the difference here is very large. So that indicates that this is more significant than the current estimate. So we increase the number of iterations, and we get a lower p-value. So now it's more definite that yes, this is a good overlap. And we see that all the time we have a history here. So here, this was the first run. Here we have actually the second run, but I did a mistake, so I deleted one of them, but I could have kept it just to illustrate that we have continuous growing list of the experiments. And then we can go back and rerun things. You click on this eye to open the window and look at the result. You click on this one to rerun it, so you keep all the parameter settings. But then you can change one or two parameters and run it again. Okay, let's just for fun try a different epidemic mark. This one indicating repressed regions. And of course, as expected, we don't see an also. No support for this conclusion. P-value is 1. And this is as we should expect. So this is mainly a test of is reasonably what is done. Okay, let's try something completely different. do we tend to find SNPs associated with Parkinson's disease in active promoters? And I have to admit that this is a very random choice. I was using this GWAS catalog of 636 data sets. So I just scrolled down until I found something that looked familiar, and Parkinson's disease was one of the interesting ones from our point of view. But the question is sort of interesting. What are these GWAS data really associated with? If we assume that they are true GWAS data, they cover the whole genome, do we find them more frequently in gene regulation than in other regions? We are not really answering that question here. I will come back to how we can answer that more largely afterwards. But it's sort of an interesting question. Do we find these mutations or SNPs associated with Parkinson more frequently inside the segments, meaning the regulatory regions, than we would expect by random chance? Okay, so the simplistic answer is maybe there is a weak evidence, which is maybe not very surprising, but sort of reassuring. But what you should be aware of here is that the number of data points associated with Parkinson's disease is very low, it's just 67. And although what we observe as inside these active promoter regions is twice as many as we would expect by random chance, it's still sort of weak. We are, in a way, throwing away a lot of information, maybe, by looking at those that are inside compared to outside. So it could be interesting to test an alternative. And one thing we could test is to look for, are they located nearby rather than inside outside? Because also the promoter regions are sort of segments that are defined in a specific way. Maybe that is not optimal. Maybe it's not optimal for that particular cell type we are looking at here. Maybe that cell type is not optimal for Parkinson's disease and so on. So we could try to extend it a little bit. And it turns out it may be significant, at least in some regions. Here we have 11 significant pins, meaning chromosome arms, out of 27. Some are thrown out because of lack of data, but still, it's something that could be worth looking further into. Let's try a different hypothesis here. Maybe the SNPs are affecting, rather than binding inside the promoter region, maybe it affects, for example, DNA looping. And we know that DNA looping is stabilized by cohesin. And cohesin is stabilized, or RAD31 is one of the proteins that are involved in cohesin that ties together the loops in DNA looping. And if you test for that, RAD31 close to Parkinson's disease, yes, data suggests this. These are things again, 11 significant things out of 27, but it seems to be a link. But let's now test the obvious additional test. These RAD21, do they overlap with our previous test, the active promoters? And it turns out that yes, highly significantly so. So the important thing to realize here is that we actually have been testing 2 times for the same thing, although we believe that we're testing for different things. And this is what we should be very careful about when you do this type of testing, known as confounding effects. The classical example of that is that somebody published that if you're drinking a lot of coffee, then you are likely to get cardiovascular disease, or some other types of link. And disease outcome, cardiovascular disease, coffee drinking, yes, there is an association. But the real association turns out to be that if you're a smoker, as part of smoking, it's also nice to take a cup of coffee. So they tend to be heavier coffee drinker than the rest of the population. So the real link is that the smoking increases the risk of cardiovascular disease, and the coffee drinking is just associated with that. So in our case, we know from other data that chromatin interactions often happen close to promoters because they establish the enhancer-promoter interactions. And therefore, this red 21 that ties together enhancer promoters tend to be found close to active promoters. So if there indeed is an association with Parkinson's disease, then we have to do other tests to establish what is the causal link and probably go into a lab to test that, because this is not telling us that. So you should be very careful about how to do these tests. I also want to say a little bit about how to do this type of analysis. Here I focus on the enrichment analysis, but this is really an association analysis. And this is an example that I made for a presentation in the city building in Norway. So therefore I used as an example the football club that is in Bergen. And the idea is that if I go to the largest airport in Norway, Gardermoen Airport, and ask the person, are you from the city of Bergen? Yes or no? Primary test. Are you a supporter of the football club, which is then from Bergen? Yes or no? Then I expect to see a clear correlation here. So the people from Bergen, they are supporting their football club. The rest are not supporting it. They are supporting something else. Of course, there are some people who deviate from this, they have studied in Bergen, moved somewhere else or something like that. So I get this correlation analysis. Then we can do this also proteins. Is this transitive factor automated during stress? Yes, no. Does this transitive have a zinc figure domain? Yes, no. So you can associate properties of transitive factors with their role, their functional role, and that is something we will be looking at. But here is the warning, and that is multiple testing. Because remember, you had, for this GWAS data set, for example, more than 600 GWAS sets, and it's tempting to test all of them. And what do you have to be careful about that? So let's go to back to the airport again. But instead of using only about, are you from Bergen, are you supporter of the Brown football club? You ask about all the football clubs in Norway. Are you supporting this club? Yes, no. Are you supporting that club? Yes, no. And so on. And then most of them will not be significant, but suddenly that one that is almost significant. And the problem is that this can happen just by random chance, because you are actually searching inside a distribution. And the more times you ask the question, the more times you do this test, the more likely is it that at least once or twice you will be in the outskirts of this distribution. So when doing multiple tests, you have to test for it. For example, with Ferroni, which is not the best one, but this is the simplest one. We just divide the significance level by the number of tests. It's very conservative, so there are better alternatives. But the important thing is that you have to do something like that. And the reason for mentioning it is that there is some further development in genomic hyperbrowser. We already have developed something known as HyperBrowse, which can do the same type of tests, but for 3D interactions in the chromatin. So you can take the folding of the genome into account and see how often is, for example, a given transcription factor close to an enhancer region in the folded chromatin. But we're also now writing up the final version of the paper on something called G-Suite, which is a very obvious extension. We can make a list of drugs and another list of transcription factors and test these lists against each other. And that generates a very large number of tests. And then we have to use multiple testing correction in order to get reliable p-values. So far, it's implementing only the simple tests because it's very compute intensive, but we will implement more advanced tests later. And sooner or later, we will also try to make an integrated version where we can do all of this in the same version. The advanced tests on lists, taking also 3D interactions into account. So it's an ongoing process. Okay, that's it.
If anybody question, you can ask them.
Sorry, how many genomes are available in hyperbrowser?
That is a good question, actually. It is mainly used for the human genome, but I know it has been used for a few other genomes as well. How easy it is to load your own genome into it, I'm not sure about. But it depends also on what other data are available in order to make it interesting. I have only used it for a human genome, so I'm not sure about that.
So, for example, I can't use it for data of Drosophila.
Probably not Drosophila. I believe there is some tracks for the mouse genome, but I have to admit I'm not sure about it.
And another question, is there a command line analog of hyperbrowser?
Command line?
Not for Galaxy, because I've seen that it's highly dependable of version of Galaxy.
Yeah. It is, that's correct. It's actually now implemented on a previous version of Galaxy, but it is being ported to the newest version. So it will soon be available in the newest version of Galaxy. With respect to command line version, yes, for example, the example I showed on this sort of big landscape, that was done by using command lines and scripts to automate it. So that's possible, although most people tend to use it through the Galaxy.
Thank you.
Thank you for the interesting talk. I have one question. In the beginning of the talk, you have mentioned about difficulties about identification of repeats in
Yeah.
sequences. Have you some implemented tests for working with repeats in your browser?
No. The main reason for mentioning it is that it's sort of one of the standard annotations that you see in many genomes, and it can be interesting with respect to understanding gene regulation. But we have not any specific test, but of course we have individual tracks for the different repeat types. So one thing you can do is to test whether given a regulatory feature is more frequently associated with a given type of repeat. But the problem there is that to map sequencing data in repeats is by itself quite challenging. So how reliable the data are regarding features in the repeat regions can be quite variable. But in principle it's possible to test different repeat types, for example.
The kind of testing you are using is that you are assuming that there is no replicates available, for example, so you are shuffling and so on. That is good. If you don't have a real null model, then you take your surrogate null model, which is the permutation. However, I think in real life you would often have like measurements like that are repeated or on different individuals and so forth. Do you have some testing implemented which takes replicates into account?
Not replicates. I skipped the example that was in one of the publications because that is a feature that is not very much developed yet. But that makes it possible to compare different experiments, so that you can have a normal experiment and then sort of a biased experiment and compare the differences between those. So that's possible, but to compare replicates, there is no real feature for that yet.
More questions? We have finished.
If there is no more question, then probably we will finish this session and go for lunch somewhere here. And it's going to be kind of a tricky thing because today is a holiday and I haven't thought about that. Yeah, but there should be something. And then we will start according to the schedule at 3:30. Oh, no, sorry, 3. So now it's 1:30, so it's an hour and a half for lunch, and then we are coming back.